Hi everybody. Do you? I don't think this thing works. Is it? Okay, I will just try to speak loud. So, hi everybody. It's an honor and pleasure to introduce you, Jonathan, today. Allow me to tell you why I have this pleasure today. I will try to be brief because Jonathan has a lot to tell you. So I will go back a bit in my master's years when I was doing a master in oceanology, and during my different classes about chemical oceanography or physical or biological. Anyway, I had a lot of papers to read, and you quickly associate a field of research with a name. For example, if I tell you fisheries science, you will tell me Daniel Pauli, for example. Or if I say you shark behavior, you will tell me Eugenie Clark. And well, the name I associated with marine snow in particular was this J. Trent. And from those great scientists that inspiring you in your early career, Jonathan was one of them. And imagine how surprised I was to meet Jonathan in the middle of this huge city that is La Selva Beach, to come across Jonathan at the Free Food Church, from where I bring you all these organic produces. And so when he introduced himself, it didn't take me more than 30 seconds to ring a bell and realize that I was speaking to one of these great scientists that inspired me during my early career. So I was really happy to meet him, and I'm really happy today to introduce him to you. And now I'm handing over to Jonathan. Thank you, Jonathan, to be here.
Thanks, Martin. Well, so I haven't given a talk in a while, but Ivano and Martin were suggesting that I give a presentation to you about the work I've done in various fields, and I was trying to think, what should I talk to you about? I mean, you may know me perhaps from the photographs that are in the hallway that have been here for 20 years and become part of the wall. And but those photographs, as you'll see, were part of my legacy. I didn't actually work here, but we'll talk a little bit about where I have worked. I decided when I was thinking about what I would like to talk to you about was to present three stories, three lessons from my career that start with the marine stow studies that I did here using scuba, and really represent a kind of scientific perspective. And then, if there's time, I'm going to go on with hydrothermal events and the limits of life. And that really teaches us about the scientific process. And then finally, if there's time at the very end, I want to tell you about my research on extremophiles and what extremophiles teach us about cell biology, and that's a real lesson in science personalities. So, starting with a marine stow story, can we turn the lights down? I don't know if that's possible. Oh yeah, great. So the marine snow story started with John Pierce, subtitled Ecology course in Hopkins Marine Station studying the kelp beds. And it continued with Mary Silver when I took a pelagic ecology course as an undergrad at UCSC. And later on, when I went to Scripps, it involved a guy named John Isaacs. So Mary actually gave a talk about marine snow and the odd history of a ubiquitous ocean phenomenon over 10 years ago here at Moss Landing Marine Lab. So the people who were involved in in the course with John Pierce included Alan Shanks and John Madison, among others. And later on at Scripps, Mark Olson was a big player in the whole open water diving story. And all three of these guys have gone on to have amazing careers. In fact, Mark Olson now looks like this, and he runs an organization called C-SCAN. He's also founded a company called Deep Sea Power and Light, and there's 300 people working in his company called C-SCAN. John Madison went on to become an MD and has now a scientist in residence at UC San Diego, has done a huge amount of work at Kaiser Permanente, amazing character, and works on X Prize. And Alan Shanks, who I'll talk about a lot, Alan taught marine biology at the University of Oregon for years and years and years. So what I thought I should do for you is give you a feeling for what actually happened. So here are two scientists, or two young scientists, undergraduates, who have been diving in the kelp beds and off the coast of Hopkins Marine Station. And we've been studying now pelagic ecology. We went to Mary Silver, who was the professor in the course, and we said, you know, we're divers. Do you think we could go out and look in Monterey Bay and see something? And she said, well, I don't know why not. And she gave us the keys to the Boston Whaler. And we actually took the Boston Whaler, and if this sounds crazy, it really was, and we went offshore for an hour, way offshore into Monterey Bay, and we reached a place where we're out, we can't see the land anymore, and we put on our diving gear, and we went in the water. And I mean, you know, Monterey Bay, the bottom was like probably 300, 400 meters below us. And it was dark, and it was, I mean, we got in the water, and it was not completely clear, and we could see each other, and we were sort of looking around to see if we could see anything. We don't see anything, but we noticed that our ears are popping, and as our suits are compressing, we're sinking. And all we can see is each other. And we thought, oh my God, we're going down. Our depth gauges are saying we're getting deeper and deeper. And we start signaling to go up, and we're getting a little bit scared at this point. And we're coming back up, and we're swimming, but we can't really tell we're even moving because all we can see is each other, and we're both kicking, and we're seeing each other, and we don't see much. But we're looking around, and we're trying to get back to the surface. And during the time as we were coming up to the surface, I remember seeing this weird jelly organism. And I had no idea what it was. And I think it actually wasn't Bolenopsis, which is shown here, but it was a large ctenophore called Leucothea that comes in Monterey Bay periodically. And it was just floored me. It was like, wow, what is that? And even though we were both freaking out because we thought we were going to die, I remember this image of this organism totally, profoundly influenced me. And when we got to the surface, there was no boat. The Boston Whaler had drifted a long way because while we were underwater, of course, the windage on the boat, we weren't attached to the boat. And so we got to the surface and we're going, oh shit, what are we going to do? And so we swam over to where the boat was and we hauled out of the boat, and we were both sort of desperate. And I remember the first thing I said to Alan is, did you see that? And of course I was referring to this huge jelly ctenophore. And so the next step was obviously to we could do this without dying. And we quickly evolved a system where we attached ourselves to the Boston Whaler, and we put a line in the water. And shortly thereafter, we figured out that we can attach all of ourselves to the, and we got our other dive buddies, and John Madison and Tom Aiken and a guy named Otto Orzik. All of us started diving offshore. And we would take the Boston Whaler out, and we'd put this line in the water with a heavy weight, and we would attach it. And we always had a person at the junction of the lines, which we referred to as a shark diver. And they used to carry a weed puller to fend off those sharks that came in. And actually in the bay here, we saw a lot of blue sharks. And they would circle at a distance, and they should chase us out of the water occasionally, but they really weren't such a big deal. So occasionally, not always, but occasionally, we would dive in the water, and we would be in what is a veritable snowstorm. And these were large particles that we could see underwater and film underwater. And the amazing thing about this was that sometimes they were ubiquitous, and sometimes there was nothing there, right? And we decided that this was an interesting phenomenon, and the more questions we asked to the people in the marine world who should know about such things, and the fewer answers we got, we started realizing just how exciting this could be. We developed a system for photographing them, quantitative, so we could get size. And we sampled them. This is a picture of Alan sampling with a tube that had rubber stoppers on both sides. So we would slide the tube over one of these fragile particles, and we would stopper it on both sides and put it in a bucket and bring it up. We measured the concentration using a flow meter and a flashlight on a little loop or a bigger loop, and we would swim around on our line and count the number of particles passing through that loop. And the flow meter would tell us, how much water we had traversed. And we saw these particles on occasion, usually, when there was pretty good algae blooms going on. And we published a paper this is 1978, but that took some years before it went into press, in situ and laboratory measurements of macroscopic aggregates in Monterey Bay, California, in limnology and oceanography. Mary Silver was on that paper, and then Mary and Alan and I also published a paper on marine snow in Science, because Mary said, oh my God, this stuff is really exciting, and we put together a paper, and as you see in this photograph, there are diatoms and fecal pellets, and there are exoskeleton from a copepod right in the center. And I really, really wanted to go beyond Monterey Bay. As a young scientist and seeing how exciting it was diving in Monterey Bay, I had a dream that we could go way offshore. Because obviously Monterey Bay is a tiny part of this vast ocean that we're all interested in. And Mark Silverstein, and I don't know if any of you know Mark Silverstein, is that name familiar to you? Sure. So he was here at Moss Landing back in the day, and he's now obviously the Elkhorn Slough Foundation director. But Mark found out about the training ship Golden Bear, which was a training ship from the Merchant Marines. And it made a cruise out to Hawaii, from Hawaii to Tahiti, from Tahiti to New Zealand, back to Hawaii, and then to San Diego before coming back. And Mark Silverstein, you know how he is, he organized a cruise that we could go along on this training cruise, and we would dive. And he approached the admiral of the Merchant Marine School, and he arranged for us to come along as divers. And we were going to be able to dive on this enormous cruise track. And it was incredibly exciting for us. And we thought, this will be great. We'll be able to cross major oceanic currents, and we'll really have a sample of what the oceans are like. So we came aboard, and we went off on a 2-month-long cruise. And there was one problem. The admiral of the Merchant Marine Academy didn't get approval from the captain of the training ship Golden Bear. And so when we went out on this ship, the captain said, no way, no how, you guys aren't going in the water, nowhere near the water. And so we were kind of stuck on a ship for 2 months without any opportunity to go diving. And so I decided that I didn't want to just hang out, so I made a plankton net out of a pillowcase with a jar at the end of it, and I screwed together this plankton net. And the ship didn't slow down for a plankton haul, so we were doing 15 knots, and this pillowcase was bouncing across the surface when I would put it in the water. And I ended up sampling on this amazing cruise track. I sampled the Newston. And one of the organisms that I caught in my net periodically was Halobates, shown here eating Drosophila, one of the only insects that we have in the open ocean. And when we came back after this long cruise, and I had all these samples of Halobates from these different locations, I met with a woman named Lana Chang at Scripps Institution of Oceanography. And Lana is one of the world authority on marine insects, and she was really happy that I had all these samples of halobates, or at least where they had been located around the world's ocean. And Lana was a good friend of the Institute for Marine Research, which was run by John Isaacs. And John Isaacs, his right-hand guy was a guy named Dick Schwartzlos. And Lana introduced me to John Isaacs and Richard Schwartzlos, and they asked me about what my plans were and what I was doing and so on. And I told them about this dream of blue water diving and what we had done in Monterey Bay. And they were really excited and very supportive. And they said, so what do you need to be able to do this research? And I, as a young scientist and still at that point an undergrad, I said, well, we need a grant. And they said, well, so how much money do you need? And I said, well, and I thought, well, this is Scripps. They must have a lot of money. So I said, we need $500. And the guy looked at me like I was nutty, and he gave me $2,000. And he said, okay, well, we'll give you this grant. And we were given a space at Hopkins Marine Station back then in the trailers, which were the temporary trailers behind Agassiz by Don and Izzy Abbott approved us having this space at the lab, and probably arranged by John Pierce, whose father-in-law lived across the street, and Vicki Pierce, whose father was living there. And they gave us this space at Hopkins. And we went out and we started studying. We bought a boat we called the Mola Mola, a tiny little boat, and we went out in the offshore and we sampled these particles using syringes. We would suck the water out of one of the particles and then take a sample with another syringe a few centimeters away. And we wrote a paper called Marine Snow Microscale Nutrient Patches and published this in Limnology and Oceanography. We also looked at these particles as sinking because they were moving through the water, by no means that fast, but they were sinking and they sort of look like they're sinking. And obviously everything that's associated with them would be dragged down through the water as they accumulate heavy and faster sinking things, and they drag the slower sinking things through the water column. So we published this in deep sea research, marine snow sinking rates and potential role in vertical flux, accelerating things that don't sink quickly and dragging and slowing down things that would sink very quickly, and all of which accumulates in a sediment trap, but you couldn't necessarily know what the package was that arrived. Anyway, so we've been publishing, and by this time we're getting to be known, and a guy named Bill Hamner, who had independently been doing blue water diving and mostly in really tropical areas, by now Bill Hamner has worked, he was at Davis originally, but now he's at a marine lab in Palau. And he was studying the saltwater lakes in Palau. And Bill said, why don't you guys come and help me study these saltwater lakes filled with jellies and sharks and really exciting here in Palau? And I was like, yay, let's do it. Let's go to Palau. And Bill said, well, the only problem is I don't have any money to support you. So I thought, okay, well, let's talk to the people at Scripps. Maybe they'll fund us to go and work on these tropical island ecosystem and using our blue water techniques. And I remember Dick Schwartzlos said, well, when I asked him if he would give us money to support us on this research in Palau, he said, well, I can do better than that. Whoops, sorry. Yeah, here's Bill Hamner and his wife Peggy and doing the work that they were doing in Palau. He said, we'll do better than that. We'll invite you to come to work with us here at Scripps. And you can be part of the Cal Coffee program, and we'll let you do blue water diving off our normal plankton hulls, and you'll be able to go out and off the San Diego Bight and do all this really cool research in the San Diego area. And so we went down and we did that research, and later on published with Jack Beers and Frida Reed, and Alan and I both took positions down there and went down in we're up to about I think we went down there in like 1977. And here now we're calling marine snow macroaggregates. During the time we were at Scripps, we also did cruises with people at Woods Hole, and Richard Harbison and Larry Madden invited us to cross the Atlantic with them to go from Woods Hole to the Azores, and then from the Azores to Scotland, and we were diving all the way across. And then Ken Bruland invited us to go on a cruise in the Pacific, and we went to Hawaii, and then from Hawaii to Tahiti. What you think about, that was a dream I had with the Maritime Academy, but now we actually did it in both the Pacific and in the Atlantic. And we were going out now in big ships, and we were going off these ships, and this is Larry getting ready to go off for a blue water dive, and we always took the Zodiac some distance away from the ship because the sharks are following the ships and using the waste from the ship, and we would go some distance away from the ship and then dive in the water. And now we have a line that we're diving on, and we have a program and careful system. And on the cruise in the Pacific with Ken Bruland, Ken Cole was our shark watch, and I'm sure all of you are familiar with Kenneth. And the sharks, we did see sharks. In fact, we saw sharks on a pretty regular basis, and they would usually circle at a distance in the open water when we were there. And I can tell you that fortunately this never happened. In fact, of all the times we were diving, this is just an artist's conception of what could happen. But what did happen, which was really exciting, was that we saw a lot of these gelatinous organisms, some of which are really large, like this chain of salps. Beautiful organisms, and some of these you'll recognize from the hallway. And organisms that are transparent, 40% water and 3% salt, and organisms that are incredibly beautiful to look at. These are pictures by some of my colleague blue water divers. This is Ron Larson's photograph, and another picture from Ron. And Ron took pictures also and was studying the symbiosis between the fish and some of the medusa that we saw in the open water. And Mark Olson, I mentioned earlier, he took this selfie of himself with a ctenophore called Baroe. We also saw pteropods, which are contributing to the marine snow out there with their large mucus webs that they build for feeding, like Corolla spectabilis. We very rarely saw marine mammals. There was only one occasion where I took this picture of Alan with these two Pacific whitesides that came screeching up to us and turned away and disappeared. Just long enough for me to take a picture with the Nikonis camera. Even when we were diving in the open ocean, we occasionally ran into these really high densities of marine snow. So there are some areas in the open ocean that are highly productive, although most of the places we were diving, the water was really, really clear. And so this, if you see what the water looks like here, it was completely transparent. The only thing that you would occasionally see floating by were colonial foraminifera, but virtually nothing else. And occasionally, of course, one of these jelly organisms I mentioned. So in scuba, we could sample down to 5 meters regularly, 10 meters, and even 20 meters. We could bounce dive down to 30 meters. But when you're out in the water and you're looking down that line, which is down to about 20 meters, and you see into the depths and you know the bottom is 3 or 4 kilometers below you, you always are wondering what's down deeper. And so when I was at Scripps, I had the opportunity to work with some Navy saturation divers. And a colleague at Scripps named James Orzik, who had been a diver with the Navy and was studying marine science, he got me involved with the Navy saturation divers at Point Loma in San Diego. And we went out on a ship called the USS Elk River. And the Elk River had aboard a diving submersible chamber and 6 divers, and I never did this, by the way, but 6 Navy divers would go into this compression chamber, and then they would seal it off. They would pressurize them to the depth that they were going to dive to. They would then, 3 of them would climb into this submersible chamber, seal it off, and they would lift them up and then lower them into the water. And when it got to the depth at the same pressure, it would pop open, and they would go out into the, 2 of them would go out into the water. And then they would go back into the chamber and come back aboard the ship to go back into this compression chamber. And so if you look at the Elk River, you see here's what the submersible chamber looks like on board. And this is a mobile winch that moves along these tracks. And it's lowered through this well. It's a hole in the middle of the ship. And they would go down and make dives. And so I wrote a letter to the Admiral of the Navy as a graduate student. And I said, you know, you guys are training these divers, and they're disconnecting a nut and bolt and reconnecting a nut and bolt. But, you know, they could do something incredibly worthwhile. And anyway, long story short, the Admiral of the Navy wrote a letter through the chain of command that organized and orchestrated my ability to go on these cruises. And you notice the diver has got a syringe in his hand, and he had a string of syringes in a container hanging from his wrist, and he would simply collect a sample and put it in a little bucket outside of their chamber, and they dove to 75 meters, 140 meters, 200 meters, and 260 meters depth. This is called a sliding saturation dive. They're completely saturated. They can take as long as they want in the water. Their suits are warmed by warm water pumped down to them, and they're saturated. So they can stay in the water and take samples for as long as it takes. So Otto Orzik and I wrote a paper on the use of Navy saturation divers for research, and the only catch is that in order to get back from 260 meters, it takes those 6 men living in a decompression chamber 9 days to decompress. So they showed them cartoons through the porthole, and they put food down through the hatch. But it was always still the remaining question, what's down deeper? Because the bottom depth that even where we were working, was way below the 260 meter limit. I mean, the average depth of the ocean is 3.8 kilometers. So I had the good fortune of going down in Alvin, and I made a couple of dives in Alvin. It was remarkable to go down into the deep ocean. But the problem with Alvin was just the lack of dexterity, the inability to really interface with the water very easily. And remember what we're looking at. We're looking at these incredibly fragile particles that if you swam up to them and did this, they would fly to pieces. Well, I later in my career worked with NASA, and I realized that one of the take-home lessons here is that in the 1960s, we thought that in order to really research something, you had to go there. But you know, and this is an interesting picture because this, let's see, do I have a pointer here? Oh, anyway, the lower tiny robotic system is called the Sojourner. And I started working at NASA in 1998, and Sojourner went to Mars in 97. And the one that's up behind the 2 guys that are standing there, that's the model of Spirit and Opportunity, which went to Mars in 2004, both of them in different places. And Curiosity, which is on the right-hand side of the screen, that's the robot that went in 2012. And it's still up there. I mean, they're still collecting data. They collect data from these for a really, really long time. And so the lesson learned was that you never send a person to do a job that a robot can do better, faster, and cheaper. And so when it comes to blue water diving, we really should look to what are the techniques that are available that allow us with remote vehicles like the Ventana at Mabari, or the sediment traps that are now being used to be able to sample remotely these environments that are accessible by diving, but that are really restricted in what you can do, where you can go as a diver. So marine snow, if you look at it in this photograph that I took underwater, marine snow is part of a size continuum of particles. If you think about what's going on in the ocean with detritus, the particles run from microscopic to macroscopic and quite large. And you can ask the question, is marine snow important? And the answer, of course, really would depend on what questions you're asking. I told you about patches of nutrients. I mentioned associated organic material and diatoms and such things. But if you're a whale, it's maybe completely irrelevant to you. If you're worried about the transport of carbon through the ocean, it may be important that there are particles intercepting things. But the bottom line is really marine snow studies of scuba provide a really interesting scientific perspective, but the data without context is really useless. It really depends on what questions you're asking as to whether or not something is relevant or not. So let me shift gears here. I'm about halfway through my time. Let me just tell you a little bit another story about hydrothermal events and the limits of life. And this is really meant to give you a take-home lesson about the scientific process. I told you earlier that Alan Shanks and I published this paper about marine snow in terms of its transport through the water column. And indeed, marine snow is sinking. You could see in these images, it looks very clearly sinking. So what would happen to a microbe living associated with a marine snow particle? It would be dragged down through the water column at a rate that it would not be able to otherwise go through the water. And obviously, as it goes past the thermocline, it gets into cold water. And as it gets deeper, it goes to higher and higher pressures. So as a graduate student in Art Yanis' lab, I asked the question, what are the pressure effects and temperature range, and on the temperature range for growth and survival of a marine bacterium like Vibrio harvii, and what are the implications for bacteria attached to sinking particles? And the answer to that question is that if you think about the ocean, as you get into a really deep environment, it is cold and there's super high hydrostatic pressure. And I mean, if you look at the paper, you realize that at intermediate low temperatures, the high pressure is deadly to these microbes. And at lower temperatures, they're pretty well preserved. And that pressure doesn't influence microbes that much at all, unlike humans, which can get down to maybe 70 meters. E. coli grows quite fine at 250 meters, 250 bars, and humans go down to about 70 bars. So the ocean, the deep ocean, is cold, mud, huge, vast plains of this that are totally dark and high pressure. There are a few places in the ocean, however, where black smokers are found, places where this cold water permeates through the sediments, gets close enough to a heat source, and then is warmed up and finds its way back to the surface in the form of what is called a black smoker. Black smokers are black not because there's anything to do with smoke, but rather the metal sulfides in the water come out as superheated water, and they precipitate when they get into the 4-degree water. So the temperature, the maximum temperature for seawater is about 420 degrees Celsius. And the ocean surrounding is about 4 degrees. So we see this precipitation as smoke-like material. These black smokers, which were observed by Alvin, excuse me. Do we have a cup of water? Yeah. these black smokers are surrounded by life. I mean, vestimentiferous worms, and we see crabs and all kinds of high concentration of life forms that are surrounding these black smokers that are on the bottom. And the temperature of one of these black smokers was measured to be about 300 degrees Celsius at 2,650 meters depth, which is about 265 bars of pressure. And the person who took these samples was a guy named John Barrows, who at the time was at the University of Oregon. And John came to our lab at Scripps with samples from this black smoker, 21 degrees north off the coast of Mexico. And he asked us to build equipment for him that would allow him to simulate the growth conditions of high temperature and high pressure. In order to do this, we built this apparatus. And I need to tell you a little bit about how this apparatus works in order to tell the rest of the story. So bear with me. But the reality is that we could put a titanium syringe into a container, a pressure chamber, and we heated it with silicone oil, and we put a sample inside the titanium syringe. And that spherical structure in there is a ball bearing. And we would tilt this thing back and forth, and the ball bearing would roll back and forth, and it would mix the sample. And there's a piston. When we wanted to take a sample, we would put silicone oil into the back of this chamber, and it would displace that piston and push the liquid through that valve and out into this, with this pumped material, out into this cooling chamber, which also allowed us to decompress the sample. So everything's at high temperature and high pressure, and then we shift the sample by injecting silicone oil, displacing a little bit of sample with that piston moving, and it goes out into that other chamber, which is also high pressure and high temperature. We then isolate it with the valves, we cool it down, and then we can decompress it and remove the sample. So this is important because we did these. John Barrows, working with a postdoc in the lab named Jody Deming, did these experiments using this apparatus. And that cooled sample was what they ultimately got out, and that allowed them to take samples with time of specimens that are inside that titanium syringe. Now, John and Jody, John was the visitor to the lab, Jody was a visiting postdoc in the lab from Johns Hopkins. They submitted this paper to Nature in 1983, and it really was a wow. They claimed to find bacteria growing at 250 degrees Celsius. The highest temperature organism at the time that was known grew at 115 degrees. So they were way, way, way above the temperature. And this paper was published, and it was a sensation. And you guys are mostly too young to know that this was even perhaps published, but it was a huge sensation at the time. And the reason, of course, was because the pressure, the argument was that the high hydrostatic pressure allowed us to have liquid water. And liquid water was seen as the critical element for life. So let's look at the evidence for this. The main evidence was that they showed an increase in DAPI-stained cells, which DAPI is a DNA-specific dye that when it gets inside a cell, it percolates into the DNA and it fluoresces blue. And they could show that there was an increase in these blue fluorescent particles with time that they saw were DNA-containing cells. They also showed samples from 250 degrees and 265 bars contained recognizable bacterial cells in the electron microscope. And they showed these pictures in their article. They were unequivocally bacterial cells. So I was a graduate student in the lab, and I had been working on high temperature and pressure effects on microbes. And guess what? This result was outlandish. And so I want to walk you through what I did with Roger Chastain, who was another graduate student in the lab. the first thing we did was to try to do the control experiments that John Barrows and Jody Deming didn't have time to do. And so we took in the same media that they use with no inoculum, and we put it inside the titanium syringe and heated it to 250 degrees and took samples over time. And guess what? Particles spontaneously formed at this super high temperature and pressure, and that with time, we saw that the O-rings that were in the system failed. What did that mean? That meant that the silicon oil was leaking in to the sample chamber. And every time we tried to take a sample by pumping silicon oil into the outside, silicon oil was getting pushed into the sample, and it was coating those particles. We found that silicon oil fluoresced blue. What did this mean? The increase in DNA containing blue particles, with time, there wasn't any increase with time. It was blue fluorescence on these spontaneously formed particles that happened to be about micron in size. So I remember when I got this result, I called John Barrows and I said, John, because his paper, I think at the time, had not yet been published. We had seen a preprint of it, and I said, John, I'm seeing blue fluorescent particles that are coming from the silicon oil. And he said, well, it doesn't matter because nobody has ever seen bacteria that can survive at 250 degrees at 265 bars of pressure. So in any case, we're going to go ahead and publish the paper. And it's true that when we looked at the samples that came out of that titanium syringe that were blue fluorescent, we looked at them in the electron microscope, they were totally different than the beautiful pictures of cells that John and Jody published. Those particles that spontaneously formed were minerals. And they were precipitated in the electron microscope. They looked like a mineral precipitate. But then we realized that the way you process a sample to get it into the electron microscope was different than what we had done in Oregon. And when we copied the exact procedure that they used in Oregon, the cooled samples were contaminated with bacterial cells. And that's what they were seeing in the microscope. So that there were samples, cooled samples that contained contaminating bacterial cells seen in the electron microscope, and they were never exposed to 250 degrees and 265 bars of pressure. And so Roger, the graduate student, and Art, the professor in the lab, and I published a paper called Artifactual Basis for Apparent Bacterial Growth at 250 degrees Celsius. And this was also published in Nature about a year after their paper occurred. So their results, which were really a wow, were completely wrong. So the lessons here, what is the science lesson learned? And the science lesson learned is that extraordinary claims require extraordinary evidence, which is what Carl Sagan said many years ago, and that the postdoc that was working with John Barrows should have insisted on controls, and more controls. But even more telling, in my mind, is why the editors of Nature and the peer reviewers let this paper go to press. They should have stopped this paper from being published rather than exposing John Barrows and Jody Deming to the criticism they got after their results were shown to be nonsense. And by the way, the 250 degrees is completely outlandish. I've published a paper about this called In the Realm of Phonions and Confusoa, and it was published in La Recherche and El Mundo Científico. Phonions were when scientists cheat to get results. Confusoa was when scientists don't do enough control experiments, and they get confused and they get overexcited about a result, and they see things that aren't there. What I really learned about these black smokers, and the take-home message about black smokers is that, bit like this campfire, is that it's true that there's life forms all around it, but you can't get too close. and if you get too close, you die. Okay, so I'm going to take a couple minutes now and tell you about an extremophile perspective on cell biology, if you're still with me. And this is another shift, but it's closely related. And these two guys, Wolfram Zillig and Karl Stetter, were the two people who published the 115 degrees Celsius bacteria that was blown out of the water by the black smoker bacteria, which I refer to as BS bacteria. But Zillig and Stetter were chemists in Germany, both working at the Max Planck Institute for Biochemistry, and they had found these real organisms living at 1 bar overpressure at 115 degrees to 118 degrees Celsius. And they obviously were very excited about papers showing that the 250-degree bacteria didn't really exist. And Wolfram Zillig, who was the director of a big department at the Max Planck Institute, was sampling around, but was really a biochemist, and he invited me to come and work with him, helping him in his field sampling. And I did go to the Max Planck Institute as a postdoc and went really many, many places with Herr Zillig to collect samples. And we did a lot of things together, and he, like climbing and boating, but we did a lot of sampling and isolated organisms and pyrodictium and a number of organisms were, Pyrococcus was one of the organisms that we named. But when I was in Zillig's lab, it was a biochemistry lab and a molecular biology lab, and the project that I chose to work on had to do with something called acquired thermotolerance. Now, acquired thermotolerance is the observation that if you directly expose an organism to a lethal temperature, that is, if you take all kinds of organisms, virtually even human tissue cells, any kind of organism, and you shift it from its normal growth temperature to a lethal temperature, it dies. And by definition, it's you shift it from its normal temperature to a lethal temperature, it dies. Acquired thermotolerance is if you pre-expose this organism to a near-lethal temperature for a period of time, and then you shift it to this high temperature, it greatly enhances its survival. And you can see that in the data here from E. coli, and that is if you grow E. coli at 35 degrees Celsius and you shift it to 45 degrees Celsius, it dies in the control experiment. If you heat shock that cell at 40 degrees, before shifting it to 45 degrees, it survives much, much better, as you can see in the data here. And this phenomenon called acquired thermotolerance is associated with a certain class of proteins called heat shock proteins. And so if you look at this autoradiogram, E. coli on the left, control, this is looking at protein synthesis. You can see the proteins that E. coli is making under its normal growth condition. When you heat shock it, IEHS, you see that it's making some proteins in large amounts. And one of those is a protein called HSP60. The central column is Saccharomyces cerevisiae, and the column on the right is Drosophila melanogaster. And you see that each of these organisms make heat shock proteins, this special group of proteins that they synthesize when they're exposed to high temperature conditions. If you take the heat shock protein 60 and you look at the protein in the electron microscope, you can isolate it from the cell as a ring structure shown on the bottom left, and a side view, so-called side view at the bottom right, shows you that these small ring structures are being formed. So for me, I was very excited about the idea, what would happen to an organism living at 85 degrees, pH 2, so-called Sophilobus shabbatai? What would this organism, how would it respond to growth, a lethal challenge and an intermediate heat shock? And actually, to my surprise, it had a totally classical heat shock response. That is to say, if you took the cells grown at 70 degrees in the blue and you shifted it to 92 degrees Celsius, it died very quickly. If you shifted those cells first to 88 degrees for 1 hour and then shifted them to 92 degrees, they survived incredibly well. And I published this with some graduates, with some postdocs in the lab, Joris Oshapuk and Tom Pinkow. And we looked at the acquired thermotolerance and heat shock in an extremely thermophilic archaebacterium, Sophilobus. So then the big experiment was, what heat shock protein does this organism make? And this is just a graph showing the temperature and a radioactive methionine labeling experiment. And we labeled it in the first position during its 70-degree growth, and then we shifted it to 88 degrees at 2, 3, 4, and 5, and then we shifted it to 92 degrees at 6, 7, and 8. And you make an autoradiograph of that, and to my surprise, there was a single band on a gel that was showing that one protein was predominantly being made at that high temperature shift during that heat shock period. So the problem then was finding out what that protein was. And I worked and worked and worked to it. The problem was, and this is just representing how much effort it was, I tried to get the gene for that protein by techniques predating PCR, right? And so without polymerase chain reaction, it was difficult to get back to the gene for that protein. And I did this work first in Wolfram Zillig's lab, and after a year at attempting to do this, I left his lab in Munich and went to Roger Garrett's lab at the University of Copenhagen and tried to do in Roger's lab to get the protein. There, too, I was unable to get the gene for the protein. I could purify the protein. I could get partial sequence of the amino acids, but I could never get the gene for the protein. So I moved to Paris at the University of Paris at Orsay and worked in Patrick Forter's lab, and there, too, I was unable to get the protein. I came back to the United States and worked at Yale at the Boyer Center for Molecular Medicine at Art Horwich's lab, and it was only there that I managed to finally get this protein. So we found, in fact, before that the protein formed this ring structure that was similar to what I showed you for E. coli in the bottom graphs, but instead of having 7 subunits around a ring, it had 9 subunits around the ring. I called the protein structure a Rosetta Stone because I was playing on the concept of a Rosetta Stone. When I sequenced the Sophilobus gene, it wasn't related closely to any of the bacterial proteins. So there were a lot of bacterial proteins, including chloroplast and mitochondria proteins, that had been sequenced for this HSP60, and they were phylogenetically very closely related, but the Sophilobus protein was not at all related, as indicated by the length of the line at the distance it was from those organisms. But it was closely related to eukaryotic proteins. In particular, it had an incredible identity with a mouse protein called TCP1, tailless complex polypeptide 1. And TCP1 had been studied in a ring structure similar to this, but this was a breakthrough and published in Nature, and we had a cover on the issue because this heat shock protein in Sophilobus was related to these eukaryotic proteins, and it was described as a new molecular chaperone. So very briefly, we published this paper in 1991, and as I mentioned, I was in Art Horwich's lab, and most of the work was done by me and a guy named Elmer Nimesdown in Germany. So I thought this cartoon was appropriate. You're completely free to carry out whatever research you want so long as you come to these conclusions. In other words, Art Horowitz's lab was a lab studying molecular chaperones. And I wasn't necessarily convinced that molecular chaperone theory was right. And in fact, with my further research, I showed that Sophilobus HSP60 is not a molecular chaperone. And not only is it the heat-shock protein not a molecular chaperone, but this concept that inside the cell, protein folding is mediated by a specific group of proteins called molecular chaperones, this concept is wrong. And I know you read about it in textbooks now, but this concept is a non-starter. And so you see the picture is the double ring structure, and you see the protein is folding inside this structure. But we now know the structure of the heat shock protein from Sophilobus and other organisms, and that structure for the subunit looks like this. And the protein, when it assembles into a double ring, completely fills the central cavity. So the notion that there's proteins folding, hiding inside this little ring structure, it doesn't work. But more importantly, I went on to localize the protein in Sophilobus. I went to a lab at Harvard, a guy named Losick, and we looked at where the protein was in Sophilobus. And the protein is a membrane protein, unequivocally. But think about it. Sophilobus lives at pH 2. The inside of the cell is pH 6.5. There's 4 orders of magnitude of protons trying to get in through the outside of the cell. And when you heat up Sophilobus, the membrane leaks and it kills the cell. If it makes heat shock proteins, the membrane doesn't leak and the cells survive. That was the explanation. The molecular chaperone idea, I have tried to publish papers called Sensitive Chaos as a theory for how proteins fold in vivo, but there's a pretty strong group of people that are advocating for molecular chaperones still to this day. So let me end my presentation now in a couple minutes and tell you that Percival Lowell was a very famous businessman, and in 1914 he had this huge telescope built, and he looked at Mars and he described and made maps of the canals on Mars. By 1965, the Mariner 4 flew past Mars, and there were no canals there. And now we have stunning pictures of the surface of Mars. There is no ancient civilization that made canals on Mars. And now the problem is getting the techniques we need to be able to visualize what it's like for individual protein structures inside cells. And I worked for years to try to be able to do this, and this is about as far as I got. The resolution isn't good enough quite yet. So finally, these were the 3 stories I thought I would tell you. A story about marine snow and scuba to expand our science perspective. A story about hydrothermal vents and the misguided concept of upper temperature limits of life because control experiments, because the science process failed until we corrected ourselves using the controls that we're all rigorously taught to use. And finally, the extremophile perspective on cell biology provides an external look into the system that's incredibly valuable. So with regard to the lessons in science, our perspective, I think, is stated well by Marcel Proust, who said that the real voyage of discovery consists not in seeking new landscapes, but in having new eyes. The process, I quote the motto of the Royal Society from 1662, nullis in verba, take nobody's word for it. And finally, with regard to the personalities in science and humanity, E.O. Wilson said that the real problems of humanity in science is that we as a species, we have Paleolithic emotions, medieval institutions, and godlike technology. So I'll end there, and I think there's no time for questions, but thank you very much. So we're past time, but if anybody wants to ask a question, I'm open to it. Do you want to turn the lights up? Oh. Are there any questions? You know, I didn't know what to show you guys. I could have given you a whole talk about marine snow, but then I thought, you know, marine snow is interesting, but you guys probably know more about that than I do by now. And I thought it might be interesting to take you on this other journey. Yeah.
I'm not sure what I'm asking exactly, but just because you said at the end about needing the resolution to see some of those proteins, I know there's like atomic force microscopy, but also freeze.
For what? For which one?
The molecular chaperones,
Oh, yeah.
you know, surface proteins and membrane proteins. And I wondered if anyone's tried like freeze etching with EM or something so they can see proteins sticking out on the surfaces of those.
So these proteins are made cytoplasmically, and they seem to be going to the inside of the membrane. They're not on the outside of the cell. So Sophilobus is surrounded by what's called an S layer, a surface layer, which is like chain mail. It is protein, and it is a strong structure, but it's not crucial to their survival. All organisms change their membrane chemistry when they change their temperature, and Sophilobus is no exception. But that process takes a long time. They have to go through lipid synthesis, and it changes. And I've written a lot about this. If you're interested, I can give you references. But the reality is that the chaperone concept that proteins are folding in vivo inside this little structure, it makes no sense. First of all, only tiny proteins could fit inside. Second of all, even if they did fit inside, the concentration, there are organisms that don't have HSP60 that have now been identified. So how do they fold their proteins, right? And the notion that these organisms are using these heat shock proteins to stabilize the membrane is so much more sensible, because the membrane is this 5 nanometer thick layer that separates the whole of biochemistry from the outside world, which is completely divergent. It just makes so much more sense. But the field is now way down the road of molecular chaperones, and I don't know if they'll ever turn around. I mean, it took me 17 months to publish that paper that I showed you the reference out to, and it got rejected in Science, it got rejected in Nature, and I went back and forth, and I write back to the editor, but wait a minute, these reviews are absurd, and I have all these compelling data, and they didn't address the data. And they would say, you're right, we'll send it out for more review, and more review, and more review. So it finally got published in the Proceedings of the National Academy, because I knew somebody on the Academy, and they put it in as a paper. But it's never been cited, as far as I can tell. It was basically buried. And Art Horwich and Ulrich have gone on to get the Lasker Prize for discovering the molecular chaperone concept, and they've gotten the Breakthrough Prize, and it takes us a long time. We'll correct this as scientific community, but it takes a long time when we go on these divergent paths to get rid of bad ideas. So it'll evolve, you'll see. Mark my words. Any other questions? That reference in PNAS is the best one that I think we have out there. I've written some other things about it. Any other questions about marine snow? You guys are doing marine snow? Is there any questions about marine snow experiences? Very generic concept, by the way. It comes in all different shapes, sizes, and flavors and types. And I think we have a problem as human beings trying to generalize about things and put them into language. But anyway, okay. Did you guys know about the Barrows-Deming experiments before? The experiments that 250-degree bacteria?
Oh, I knew about it.
You knew about it?
Well, John Barrows was on my PhD committee.
Oh, great. John Barrows went on to become a really successful scientist at the University of Washington and was part of the Astrobiology Institute. So yeah, I know John really well. And Jody. Yeah, they screwed up big time. But they never retracted their paper.
Yeah. I think they still stood by it.
They still do.
I think so.
So The people who pushed me as a graduate student were Stanley Miller from the Miller-Urey experiments and Leslie Oracle, who was an incredible biochemist written about the origins of life. And Leslie is the one who said, if you name this artifact, you'll be famous, Jonathan. And I wrote the paper on phonions and confusoa. But no, there's no way the biochemistry can happen at those temperatures. I mean, it's just, if it had been 150 degrees, it wouldn't be a different story. It would be wrong still because the control experiments, but it wouldn't have been so outlandish. 250 degrees, it's just, there's no biomolecules. And the pressure, even though the pressure keeps water liquid, and you could argue DNA is stabilized. You know, DNA comes apart at high temperature. You can stabilize DNA very slightly at 1,000 bars. You can stabilize it. Because what pressure does, if the chemistry goes to a larger volume, then the pressure keeps that chemistry from happening. But with DNA, you stabilize 1 degree higher melting temperature for DNA at 1,000 bars of pressure. So 265 bars, not going to do it. And life forms in those hydrothermal vents, you can put your finger through a flame, that works. But having things living at 250 degrees, no way. There was no data in any case. At least no compelling data. There might be life on Venus. We can believe it if we want, but anyway, not science.